Greetings to everyone. I'm Sivasankar A.S. from India. I'm currently pursuing my master's in the stream of biotechnology at Sastra Team 2B University, Tanjavur, Tamil Nadu. I'm glad to present my work in this conference, which is about drug repurposing-based identification of dengue virus envelope protein inhibitor to log in its dimeric metastable state. Dengue is an herboviral disease caused by dengue virus, which belongs to flaviviral family, and mosquitoes act as a vector in this disease. This is being an epidemic in tropical and subtropical countries for last 2 decades, but there is no drug or vaccines available for its treatment. In the life cycle of dengue virus, the envelope protein E, which is associated to protein C and protein M, plays a significant role in the attachment of the viral cells to the host cells. The viral and host cell fusion occurs due to a conformational change in this envelope protein at a low pH environment. This conformational change is from a dimeric pre-fusion state to a trimeric post-fusion state. This conformational change causes the entry of dengue virus into the host cells during the course of infection. Thus, the objective of this study is to log the envelope protein on its dimeric metastable state. In a previous study, 2 novel cavities were characterized and validated, and one of which is in the dimer interface. And around 6 amino acid residues, including lysine 246, is identified to be a part of the T helper cells epitope. Thus, we have used around 3,805 FDA drugs as an initial candidates to identify a potential drug molecule to inhibit the dengue virus envelope protein and lock it in its dimeric metastable state. To perform this study, we have used or followed a sequential methodology in which the 3-dimensional structure of the protein is extracted from the PDB, and the PDB ID is 10KE. The 3,805 drug molecules were extracted from PubChem database. A preliminary docking is performed in orthodox VINA software, and good binders were shortlisted based on their binding affinities and interactions. The good binders are further subjected to enrichment docking in orthodox software, and these top 100 good binders were also clustered in Schrödinger software. The molecular dynamic simulations of these complexes will be performed in GROMACS, and a focused library of drug molecules that can inhibit the dengue virus envelope protein and lock it in its dimeric metastable state will be constructed in iLib divers. As in results, after the preliminary docking, the binding affinity of these 3,805 ligand molecules to the protein was in a range of minus 11.3 to minus 1.3 kilocalories per mole. But the good binders that are shortlisted based on the binding affinities and the interaction residues, which were analyzed using LickPlot Plus software, were in a range of minus 11.3 to minus 9.1 kilocalories per mole. These top 100 good binders were further subjected to clustering in Schrödinger platform on a settings of Tanimoto coefficient. As a result, the cluster 4 were possessing around 72 molecules, and cluster 6 was with 19 molecules. These 100 molecules, when were subjected to enrichment docking, there was no fold increase in the binding affinities. So other interactions were analyzed. The protein ligand interactions were analyzed using protein ligand interaction profiler, an online tool. In this analysis, various interactions like hydrogen bonds, hydrophobic interactions, salt bridges, halogen interactions, pi-cation interactions, and pi stacking were analyzed, on which the top 5 molecules, which including oxaquin, chromidopine, cebremostat, casopitant, and zopolrestat, which were having lowest binding affinities and maximum number of different types of interactions along their bindings to residue lysine 246. Thus, I conclude this study by saying these 5 molecules will be a potential drug component to inhibit the dengue virus envelope protein on its dimeric metastable state. We are yet to perform the molecular dynamic simulations and construct a library of these drug molecules using iLink divers.